The National Cancer Institute recently set up 12 centres around the country to develop new ways for physicists and cancer biologists to work together to tackle cancer. In his lab at the Biodesign Building at Arizona State University, Professor Stuart Lindsay and his team are examining the fundamental mechanism whereby DNA unravels in the nucleus of cells to allow genes to be expressed and translated into the manufacture of proteins vital for normal growth and maintenance. But if the unravelling goes wrong, the result may be cancer. Stuart, tell me what your project is about.
So we are looking at the chemical modifications of the proteins that wrap DNA up to control gene expression. The reason this is important is lots of people have gene defects that can lead to cancer. Very few of them actually get cancer. So cancer is not a disease based on gene defects per se, but rather it's based on the chemical factors that control gene expression. And those are the factors we want to probe.
Genes are segments of DNA and can only function when they're exposed to molecules in the cell that help to make proteins. But because the DNA is so tightly wrapped up, becoming exposed can be tricky. There's approximately 6 feet of DNA in a cell nucleus, so to fit, it has to be packaged very cleverly. It loops around groups of special proteins called histones to form connected packets known as nucleosomes. The line of nucleosomes then condenses into a fibre called chromatin, and after a lot more twisting and folding, it's called a chromosome. On Stuart's computer is a picture of chromatin, just around a few millionths of the amount found in a single cell nucleus. So what are we looking at here, Stuart?
We're looking at an atomic force microscope image of chromatin spread out over a flat surface. And chromatin's a combination of bare DNA carrying the genetic code, and DNA is this little piece of spaghetti that you see along here, with a group of proteins called histone proteins that wrap it up into nucleosomes, and these are the little white blobs you see along here.
One of Stuart's assistants is Parminder Kaur.
That's a core of the histone, and this is the DNA all winded over the core of this 8 units of the histone there.
The team is studying the interactions between DNA and the different types of histone proteins in the nucleosomes to understand how and why genes are expressed.
So using images like this of individual molecules of chromatin, i.e. the DNA plus the factors that control gene expression, we hope to see how all these various modifications conspire together to either express a gene or suppress a gene.
And as we've mentioned, gene expression depends on the physical configuration of the DNA, its packing and unpacking. So what can this tell us about cancer?
Now, well, that's a great question. So chromatin inside a cell nucleus occurs in regions where it is very tightly condensed and in regions where it's floppy with lots of accessible DNA. The regions of accessible DNA are the regions of gene expression, so it is actually this physical structure of chromatin that controls the phenotype of a cell. So in a cancer cell, presumably this pattern of squished together highly packaged DNA and floppy open DNA is different, presumably. But that's what we hope to find out with direct atomic force microscope imaging at some point.
Stuart's collaborators at the Fred Hutchinson Cancer Research Institute in Seattle are expert at extracting chromatin in a very precise way from tissue donated by cancer patients. They then send the material to ASU for analysis. It's cutting-edge work. Has this never been tried before?
The tools haven't been available to do this. So just the recognition of the types of chemical marking that control gene expression has only existed really in the last 10 years or so, and the tools for identifying it at the molecular scale are very sparse, and in fact there are none at all at the single molecule level. So for us this is a completely new development.
And then you've got to somehow correlate it to what's happening in the genes and what they do.
Precisely. But ultimately the hope is that when we look at cancer cells, we'll see what it is over and above the genomic damage that is known to exist that marks a cancer cell as a cancer cell. Because once again, you can have all the most lethal genetic mutations and not get cancer. So there are other factors over and above the genome that may be more controllable that we can discover using this technology, and those would open new paths to therapy for cancer.